In this video, we'll be talking about strategies to prevent implant-associated infections. Our learning objectives are, first, to understand strategies for preventing implant-associated infections, and we're going to reinforce these strategies with real-world examples. And then our second objective is to understand methods for testing antimicrobial efficacy. Based on the previous videos, you should now have a good appreciation for the fact that despite our best efforts to sterilize medical devices, implant-associated infections still represent a significant clinical problem. Now we're going to talk about strategies to prevent infection, which we can group into two areas. The first is anti-adhesive strategies. So in these approaches, a device coating is applied as a first line of defense against bacterial colonization. So as the name implies, we're resisting bacterial adherence. And this approach is attractive because it can be effective against a broad spectrum of bacteria. The second type of strategy is antimicrobial. And here we're talking about local administration of antimicrobial agents rather than systemic. And local administration is attractive because you're delivering a smaller amount. And then also here, we just want to protect the device. We don't really want to treat the surrounding tissue. So we're just trying to prevent infection at that implantation site. Now, other considerations are, if we're going to use these antimicrobial agents, there is concern, of course, about antibiotic resistance. So there's a lot of interest in using non-traditional agents that aren't typically used to treat infections. An example of a non-traditional agent is silver, which exerts its killing effect or antimicrobial activity by binding to and destroying bacterial cell proteins. When designing antimicrobial materials, there are several considerations. One is that we want to have long-lasting antimicrobial activity. Two, we want no inactivation of the antimicrobial by host factors. So that's inactivation due to the biological response after implantation. If it's a device that has internal and external components and we're going to coat it, we've got to coat both in order to be effective. So that pertains to like a vascular graft, for example. We want to generally try to use antimicrobial substances that are not the primary or first line of defense in other therapies against infections. And that's because of the potential for antibiotic resistance. Also, in some cases, we're trying to fight infections from antibiotic-resistant bacteria, which we'll show some examples with that in a little bit later in a few slides. We also want to ensure that there's no or low toxicity or allergy potential. So some antibiotics, like amoxicillin, for example, function as haptens, and people can have allergic reactions to those. And then lastly, we want no negative effects of the antimicrobial compound on the properties of our biomaterial. So typically they're being mixed in with polymers, so that's what's illustrated here. You're not going to be able to add in an antibiotic to a metal or ceramic, but we want to make sure in the case of polymers that there aren't deleterious effects, particularly on the mechanical properties of the polymer. Now, let's jump into some examples. For this first one, I'm going to show you a YouTube video on an antimicrobial hydrogel system.
Bacteria can live as individual cells themselves, kind of just spreading out and dividing, or they can live in an extracellular matrix, which would be that plaque that you'd find on your teeth. That would be considered a biofilm. And one of the difficulties in treating different things that have biofilm is the ability for antibiotics to penetrate into the biofilm. So one of the things that we're working on here at IBM is trying to figure out how to design materials that will penetrate into and disrupt biofilm and kill bacteria. We're taking something that is over 90% water and only less than 10% active ingredient, and we're able to disrupt the biofilm using a very mild biodegradable material. If you were to put it on your skin, you wouldn't find any irritation.
We can apply a lot of our technology materials to, Foley catheters, to, bed rails, to potentially creating hydrogel materials that are injectable, such that we could inject straight into an abscess, so a macromolecular drug, if you will. can see real world application and how you can actually affect people's lives.
Okay, so obviously that video was a little light on details with regards to the material and its composition, but I like it because it reinforces some of the concepts we've already talked about and the challenges associated with biofilm formation in particular. Also, the hydrogel that was described there was an example of an antimicrobial material, and also it was an agent that's not typically used to treat infection. So they didn't tell us about the composition, but it was obviously something inherent about that hydrogel that was antimicrobial, so it didn't contain an antibiotic. Now let's talk about some other examples. So here, these are 3 examples of polymers that have been designed to release antibiotics. So on the left, we have this Tyrex mesh. So this consists of a polypropylene mesh that a medical device can be encased in, so it's kind of like a pouch. And then this polypropylene has a coating around the outside. So this coating, if I recall correctly, is a tyrosine-derived polycarbonate. That's not important. What is important is that it has this antibiotic in it. So it's a polymer antibiotic blend. The other 2 examples on this slide are similar. So up at the top, we have these Vicryl sutures. So these are polygalactin based. So that is PLGA, of course. And so that has an antibiotic blended in with it. And in some cases, they coat it on the outside as well. And then the last one is the Simplex P. So this is a bone cement. And again, it's mixed with an antibiotic. So the antibiotic here is tobramycin. And you can see here it contains 1 gram of tobramycin. Well, let's talk about this example a little bit more. So here what I've pulled up is the 510K summary of safety and effectiveness that was submitted with this Simplex P bone cement to the FDA. So we can see a little bit more about its composition. So one thing we see down here at the bottom in the device description is it's a radio-opaque bone cement pre-mixed with antibiotic. And we see again our antibiotic is tobramycin. And here this is on the right, so this picture is showing where this bone cement is implanted. So this is used with orthopedic implants for fixation. So here this is showing a total hip replacement implant. And you see the bone cement is around the 2 components. So we have the femoral stem, and then the cement is kind of gluing it in place. And then also up top on this acetabular cup, the cement glues it up into the hip, so attaches it to the pelvis. Now, acrylic and, well, cement is a little bit of a misnomer here. So I hear cement, I think ceramics, but this is not a ceramic at all. It's actually a polymer. So it's polymethyl methacrylate, which is shown up here. So in this case, what they do is they have the polymethyl methacrylates and a little bit of monomer and some initiator components, and they do a polymerization of this. The antibiotic, again, is mixed in. It actually forms kind of like a dough consistency, and then they pack it into this syringe-looking device here and use that to inject it into the bone, and then they stick the implant and it holds it there in place. The reason this is important is because of the potential for deep infections. So, and this could even be late infections, so many, many years after implantation. So this paper that I pulled, so this is from a Norwegian study, but you see they have lots of hip implants that have been studied here. Deep infections again. They were looking at uncemented total hip arthroplasties versus cemented total hip arthroplasties with and without antibiotics. And they concluded in this study, so they were looking at almost 100,000 cases here. So they concluded that the uncemented arthroplasties and then the ones with plain cement without antibiotic had a statistically significantly higher risk of revision due to infection. So the infection leads to implant loosening, and then the implant has to be taken out and they have to redo the surgery. So without antibiotics, if we just look at this comparison with the cements here, so without antibiotics, the risk ratio was 1.9. If there's antibiotic loaded in the cement, it was lowered to a risk ratio of 1. I don't know exactly how they calculated that, but in their statistical analysis, they said the p-value was less than 0.001. So it's a very significant difference between these in terms of, so it's a very real effect of the antibiotic. So that was an example of loading antibiotics into a polymer. Now I want to talk a little bit about preventing adhesion. So one of the things that you would probably think of first is, well, let's prevent protein adsorption onto the surface. We can pegylate our material. And that's true. That is one potential strategy. But in this example, I actually want to talk about topographies. So this is from a company known as Sharklet. So it was a startup company that sort of spun out of research at the University of Florida. And their approach was they wanted to put a micro pattern on materials that mimics the pattern that's on the scales of shark skin, because that has been found to prevent adhesion of microorganisms. So this is an example of a study on their work. Down here you can see this was silicone. So we're talking about PDMS, polydimethylsiloxane, and here you can see this kind of diamond shapes that's made up of these little pillars or ridges, and you see there are grooves there. So that design is very intentional. They looked at a lot of different designs as they developed this concept. But their idea was they wanted something that would work without promoting antimicrobial resistance. So they didn't want to release an antibiotic. They wanted something that didn't rely on that. So they looked to topographies, as is noted here, and they have found in other studies that these can control adhesion. So now I want to show you a few results from their study. So this is looking at biofilm formation onto these patterned surfaces. So we have micropatterned on the bottom, and then the unpatterned is controlled polydimethylsiloxane on the top. And they looked at 3 different bacteria. So up at the top, we have MRSA, methicillin-resistant Staphylococcus aureus. So that's an antibiotic-resistant bacteria. And then on the 2 right columns, they have Pseudomonas aeruginosa. So these are 2 different strains. So the differences between those strains is not important here, but what I do want you to take away is that they're looking at 2 clinically very relevant microbes here. So these were on our list of common offenders. And what we see is up at the top on the unpatterned substrate, there is lots of bacterial adhesion and growth. Whereas on the bottom, on their micropatterned surfaces, it's drastically reduced. So we have reduced adhesion and growth of the bacteria and reduced biofilm formation. Lastly, I want to give you an example on the use of silver and how that can prevent bacterial growth. So if you've looked around in a pharmacy, you've probably seen there are various over-the-counter products that have silver in them, and they promote it as an antibacterial agent. So there are some Band-Aids and dressings and things that have it. There are also wound gels that incorporate silver as an antimicrobial agent. But here I'm going to show you some data from a study where they were looking at cotton gauze that has this antimicrobial property, so it's loaded with silver. So here, I'm not going to explain the materials very much or the synthesis, but this was a gauze and it was impregnated with silver nanoparticles. So up at the top, we actually see a little surface characterization data here. So they wanted to verify that they had silver, which is good. So this ties back into what we learned about surface characterization strategies. This, they kind of mislabeled it here. They call it electron dispersed X-ray analysis. But this is energy dispersive X-ray spectroscopy. Which you should remember about that technique. So this is the one that's always done in tandem with SEM. So you hit your sample with an electron beam, and then X-rays are emitted back, and then you can measure the energy of those X-rays and then determine what element they came from. So what they see here, so each little red dot is a point where they were collecting data from, and then they see lots of carbon, lots of oxygen, but then down here, a little bit of silver. So they say that they had 2.8% silver. So it kind of poses the question, why is the signal so low? Something for you to think about. But really, you don't need a lot of silver for it to be effective. Lastly, after they verified that it was there, they also did some studies to look at how well it inhibited bacterial growth. And so what I want to point out here is that they tested efficacy using Escherichia coli as their gram-negative bacteria, Staphylococcus aureus as a gram-positive, and then Candida albicans as a fungi. So again, they're testing against our common offenders that we know cause implant-associated infections. And then here, their metric. So this is just a measure of growth in solution. So as these microbes grow, the optical density increases over time. And so you see with the bacteria just in PBS, these black dots, there's lots of growth over time. And then with a blank cotton gauze right here, still lots of growth, not much different. They don't have any error bars here, so that's not clear if they repeated it, but that's beside the point here. But we do see that when they have their gauze with their nanoparticles in particular, so these two down here, so these have silver nanoparticles in them, there is virtually no bacterial growth. There were also some other things here, so they had gauze with antibiotic in them, and their gauze material also had some chitosan, which is antimicrobial. But here, I want you to take away the importance of silver, but then also the testing approach, where they're testing against these common microbes that cause infections. On this final slide, I want to talk a bit about how we can go about performing tests of device modifications to determine whether or not they are effective at preventing infections. So first and foremost, any testing that we do should be performed with relevant microorganisms. So by that, I mean we should be doing our tests using the microorganisms that are common offenders when it comes to device-associated infections. So the previous examples that we talked about were pretty good in that regard. Next, as we think about testing, there are kind of two questions that we can ask. So one question is, does the modification prevent microbial attachment, growth, and biofilm formation? The other question is, does the modification kill microbes? So for the first question, we really just need to image the surface. So we can do this with scanning electron microscopy. And if you think back to a lot of the example slides earlier, scanning electron microscopy was the method that was used to visualize bacteria on the surfaces. Alternatively, we can use fluorescence microscopy. So in the study on Sharklet, they used fluorescence microscopy to look at bacterial attachment and biofilm formation. So to do that, you just need a fluorescent stain to label the bacteria so that they appear. To answer the question about whether or not our modification kills microbes, there are a variety of approaches we can take. One is we can look at viability of the cells using a live dead staining method. So the dyes used would not be exactly the same as what we learned about when we discussed cytotoxicity testing, but there are comparable dye systems that can be used for microorganisms. Next, we can quantify the number of colony-forming units. So this could also be done to look at how many bacteria are present on the surface of the material. So we previously learned about colony-forming units when we were talking about sterility assurance levels. And the basic idea behind this assay is that every colony that forms comes from one bacteria and is the product of many cycles of cell division. Last, we can quantify the effects of a biomaterial on microbial growth. So this can be done in solution, similar to what was done in the last example with the cotton gauze with the silver nanoparticles, and we can use optical density to assess bacterial growth. Alternatively, we can do it with bacteria that are growing in a petri dish on top of agar. In this case, we would lay our material over the top and look at a zone of inhibition around the material. So this concludes our lecture material on implant-associated infections. Next up, we'll be learning about pathological calcification of medical devices.